Hello everyone, good evening and welcome to this NPTEL Week 7 live TA session for the course Cell Culture Technologies. My name is Ankita De and I'm one of your TAs for this course for the semester, and I host this session every week on Tuesdays 7 to 8 pm. So as you already know, this is a live interactive 1 hour video session where we discuss some of the previous year's assignment questions that help us in solving this year's assignment questions. Towards the end of our session, we have a Q&A segment where you can ask me your queries by posting them in the chat box, and I shall try to answer them. At the end of each lecture, we also provide you with the course materials and the necessary PowerPoint slides and the class recordings for your access later. So let's get started. Now, before we move on to discussing the assignment questions, let's quickly have a overview of what we have learned so far. So, in continuation with our previous video lectures, where we were discussing about the different techniques used to separate cells of interest from a heterogeneous mix of cells, we had discussed about three main techniques. So, one of them was the density gradient centrifugation method. Next was immunopanning, and the last one was fluorescence assisted cell sorting, or FACS. So we have discussed about what density gradient centrifugation is and how it is done. Now in this lecture, in our upcoming slides, we will discuss about the methods immunopanning and fluorescence assisted cell sorting. Next we have discussed about the few important considerations that one should have in mind while doing a primary culture of specialized cell types, such as the neurons or the skeletal or cardiac muscle cells in an in vitro condition. So earlier we had seen the isolation and culture protocol of pyramidal neurons from rat embryonic hippocampus, and we had used this model to study or use this model as a test bed to study the basis of disease, which is the Alzheimer's disease. So further this week we have seen about the isolation and culture protocol of the skeletal and cardiac muscle cells. So both these skeletal and cardiac muscle cells are specialized and terminally differentiated cells. And as for the culture of skeletal muscle cells, we generally use the satellite cells obtained from the adult rats, which are the main source of the skeletal muscle cells in culture, and which are then made to grow in culture and resemble their original phenotype uh, in their morphology as well as their function. As for the cardiomyocytes, they are mainly isolated and cultured from neonatal mice or rats, or it could also be obtained from the adult mice or rats. However, depending upon the source, that is where we are getting our cells from, whether the adult or neonatal rats or the fetal rats, both have their own advantages and disadvantages. As for the neonatal or the mice or rats, they have this advantage that they could go through the rapid de-differentiation cycle and are able to give rise to spontaneously beating cells quickly, such as maybe within 20 hours of plating. As for the adult cells, they also have their own advantages, such as they can they can proliferate for a longer time in culture. So there are many advantages and disadvantages of using a particular model. Now, depending upon your requirement, you can choose that which model you want to use in your study. Next, regarding the skeletal muscles, we have discussed about the different characteristic features of skeletal muscles, and in relation to that, we have discussed about the sliding filament theory, which is a theory that is used to explain the mechanism of muscular contraction of the skeletal muscle cells. So we will see in details what this sliding filament theory is in our upcoming slides. Next, we had discussed about the ryanodine receptor, the sarcoplasmic reticulum, which are also associated or connected with the skeletal muscles. Next, we have discussed in length about the skeletal as well as the cardiac cell culture protocol. Uh, And last, we had learned about how we can characterize these specialized cell types in our culture model. So moving on, so regarding the cell separation techniques, as I was saying, that we had discussed about the density gradient centrifugation, which is used to separate cells from a heterogeneous mix of cells depending upon their size and density. In a density gradient media, which is generally formed with the help of sucrose, optiprep, or nicodens media. Now, there are some other methods also that could be used to separate cells, which when the cells are of roughly the same size, however, they might express different receptors or markers on their surface, which can be used to separate them from one another. So, based on the markers, we can separate the cells, and we can use two techniques for that. One is immunopanning, and the other one is FACS. So immunopanning and in immunopanning, antibodies are absorbed to the surface of a petri dish, and cell suspensions are incubated in this dish to select for or deplete the cell types of interest. So here, as you can see in the adjoining figure, so we are taking the tissue, we are dissecting it out from a rat. We are mincing the tissue, and we are then digesting it with the help of an enzyme such as papain, and following trituration and resuspending, we take the cell suspension, and this is a cell suspension, the heterogeneous mix that we were talking about. And say this is the our culture plate, and we want to first um, we want to do a negative selection. That is, we want to remove the microglial cells. So we will take a culture plate, and we will absorb antibodies directed against some kind of microglial receptors, which are only expressed on the surface of the microglia. And then this is done. This is done to deplete our cell suspension of the microglial cells. Next, we will take the cell suspension here, and then we perform a positive selection of cells. That is, now we will absorb the antibodies which are directed against the cells of interest, and we will obtain the cells of interest. So this is known as positive selection of cells. So in a typical immunopanning purification, a cell suspension, from here we have taken the example of the oligodendrocytes. So it could be from either from the retina or other nervous system region, is prepared and then passed over several immunopanning dishes consecutively, with the first dish or two being used to deplete the unwanted cell type, such as the microglia, and the final dish being used to select the cell type of interest. So next, we have FACS, which is also somewhat similar kind of a technique to immunopanning. But here, as in contrast to what we have done in immunopanning, that is, in contrast to taking a petri dish on to the surface of which we have absorbed the antibodies, in FACS we take the cell suspension, the single cell suspension, and we make them, we uh, project our cells in a single file. We made them to eject through a point, that is, the cells are made to pass through a point through a nozzle, where they are interrogated with the help of several laser beams. So these laser beams will detect. So as I was talking about in my previous class, that they will either the light could be either uh, uh, scattered in perpendicularly, or that is the side scatter, and another one is the forward scatter. So depending upon the scatter of light, that is whether it is forward scatter or side scatter, we can detect the size of our cell and their granularity or the complexity respectively. Also, if we incubate the cell suspension with specific antibodies which are used to detect markers on the surface of these cells, and each of these markers or each of these antibodies are conjugated with specific fluorescent dyes. Uh, so with the help of that, we can identify our different cells. So flow cytometry combined with cell sorting is known as FACS. So here in this figure, you can see that the cells are either tagged with a fluorescent antibody or are stained and suspended in a liquid stream within a flow cell. The cells flow in a narrow stream towards the laser source. The beam hits only one single cell at a time. The incident light on the cell is then scattered forward as well as sideways. The scattered light is then captured by the optics in the analyzer and directed to the detector. So the cells are analyzed on the basis of the shape, size, as well as their internal complexity. So next, we have talked about the few important considerations that we must keep in mind while culturing our cells in a dish. So when we are performing a primary culture of cells such as that of the neurons or the skeletal muscle cells or the cardiac muscle cells. So these are all specialized cell types. Now, in order to culture them in an artificial environment that is outside the body, we need to ensure that a few important considerations are met. So one of them is that the cells that we are growing in our culture should mimic their original in vivo phenotype, both in their morphology as well as their function. So as for skeletal muscle cells, these cells are contractile in nature. So we must uh, we must also uh, we must also follow this in our in vitro culture methods. That is, we must also be able to have a model where our skeletal muscle cells are able to contract in our culture. And next, we have the cardiac muscle cells, which are also the specialized cell types. So these cells need to express their characteristic features in their in vitro culture plate, and only then we can say that we are able to mimic their original phenotype as closely as possible. Next is propagation. That is, we need to be able to propagate the cells for a longer time in our culture. So these are the specialized cell types that we have discussed about. There are the neurons, skeletal muscle cells, and the cardiac muscle cells. So there are also other specialized cell types that are present in our body, but we haven't discussed about them in the lectures. Next, we have the structure, the muscle fiber. So this is in the Jetson figure. You can see how a typical muscle fiber looks like. So each bundle of the muscle fiber is called a fasciculus, and within this fasciculus, this fasciculus, each individual muscle cell is known as a muscle fiber, which is connected by surrounded by a connective tissue, which is known as the endomysium. Now, each muscle fiber contains hundreds of organelles called the myofibrils. Now, you need to understand here that a muscle cell is also referred to as a muscle fiber. A single muscle cell is referred to as a muscle fiber because muscle fibers or muscle cells in their adult stages are formed by the uh, fusion of several muscle cells, and therefore they assume a multinucleated appearance. So, a muscle fiber is used synonymously with the muscle cell term. Now, within each muscle fiber, we find hundreds of myofibrils. Each myofibril is made up of two types of protein filaments, which are the actin filaments, also known as the thin filaments, and then we have the myosin filaments, which are the thick filaments. The actin filaments, as you can see in the figure here, are anchored to structures called the Z lines. And the region between two Z lines forms a sarcomere. So each sarcomere forms the basic contractile unit of a muscle. So within a sarcomere, the myosin filaments overlap the actin filaments. So these are the myosin filaments, the thick filaments, which are marked in blue, while the red thinner filaments are our actin filaments. Now, within a sarcomere, the myosin filaments will overlap the actin filaments. The myosin filaments have tiny structures called cross bridges which can attach to the actin filaments. And the mechanism of muscle contraction is based on muscle proteins which slide past each other to generate movement. The myosin thick filaments of the muscle fibers slide past the actin thin filaments during muscle contraction, while the two group of filaments remain at relatively constant length. So basically what happens is that these are the thick filaments and these are our thin filaments. So when a muscle contracts or relaxes, so these sarcomeres these increase or decrease in size. So what happens when a muscle contracts is that these two Z lines come closer to each other. That is, the thick filament and the thin filament will slide past each other so that this distance will decrease. So we will see how this happens. So there are no change in the length of the thick or the thin filaments, only that they slide past one another, which result in these Z lines coming closer to each other. So here we have explained the muscle contraction mechanism. So when a nerve impulse arrives at the axon terminal or a motor neuron, it triggers the release of the acetylcholine. So, if you can remember from our previous lecture, we had discussed about the nerve impulse transmission across the synapse. So, when a nerve impulse is transmitted across the axon terminal, it is communicated with the help of acetylcholine, which is a neurotransmitter. So, between the neuron, the presynaptic neuron, and the motor end plate, we have a cleft region, which is referred to as the synaptic cleft. So, as in order to reach from one neuron to the muscle, the nerve impulse needs to be propagated in the form of a neurotransmitter molecule. So here, acetylcholine is released in this region of our in this region of a synaptic cleft. Next, the acetylcholine will diffuse across the synaptic cleft region, and it will bind to their specific receptors on the surface of ligand-gated ion channels on the motor plate, and it will trigger a muscle action potential. The binding of the acetylcholine or the neurotransmitter onto the on the specific receptors on the ligand-gated ion channel will open channels in open voltage-gated sodium ion channels in the cell membrane, which will result in the inflow of which will result in the inflow of ions into the cytoplasm. So we had discussed about the nerve impulse transmission, but here the ion that flows in is calcium ion. So when acetylcholine diffuses across the cleft and bind to the receptors in the motor end plate, it will trigger a muscle action potential. The acetylcholinesterase which is a neurotransmitter degrading enzyme in the synaptic cleft region. It will destroy the neurotransmitter. So another muscle action potential does not arise unless more acetylcholine is released from the motor neuron. Next, the muscle action potential traveling along the T tubule will open calcium release channels in the sarcoplasmic reticulum. So, within the muscle, the endoplasmic reticulum are referred to as the sarcoplasmic reticulum, which will allow calcium ions to flood into the sarcoplasm. So, in this in a state in an unstimulated condition, the calcium ions are sequestered within the sarcoplasmic reticulum, so that the intracellular concentration of calcium ions remain very low. However, when it is stimulated, the calcium ion channel within the sarcoplasmic reticulum will open, and it will release the calcium ions into the sarcoplasm of the muscle cell, which will result in the calcium ion binding to troponin on the thin filament or the actin filament, exposing the binding site for myosin. So these calcium ions generally help in binding to troponin. So troponin in an unstimulated condition remains bound with the actin filament, thereby preventing the binding of myosin and actin filament, which is required for the contraction to happen. So when calcium is present in a higher concentration within the sarcoplasm, it will go and bind with troponin, and which will help in exposing the binding site for myosin. Next, contraction occurs, which is shown in the figure here. As you can see, the power stroke uses ATP. So ATP is required for the contraction cycle. The myosin heads split ATP and become reoriented and energized. The myosin heads then bind to the actin, forming cross bridges. Next, the myosin heads rotate towards the center of the sarcomere, and power stroke happens. Now, as the myosin heads bind ATP, the cross bridges detach from the actin. Next, the cycle is again repeated. So next, in step 7, as you can see, the calcium release channels in the sarcoplasmic reticulum close, and the calcium active transport pumps use ATP to restore low level of calcium ions in the sarcoplasm. Next, the troponin tropomyosin complex slides back into position where it blocks the myosin binding sites on the actin, and the muscle relaxes. So this is the basic pathway in which the muscle contraction happens. Next, regarding the characterization of the specialized cell types, we had discussed about different techniques which are used to ensure that the cells that we are culturing maintain their original phenotype in our in vitro condition. So for that, we can perform a patch clamp electrophysiology uh, technique, which is used to study ionic currents in individual isolated living cells, tissue sections, or patches of the cell membrane. Is This technique is especially useful in the study of excitable cells such as neuron, cardiomyocyte, muscle fibers, and pancreatic beta cells. Next, we have another method of characterizing the skeletal muscle cells, which is known as ryanodine receptor labeling. So, ryanodine receptors, as you have already seen, present on the surface of the sarcoplasmic reticulum, which are the calcium ion channels, and antibodies directed against this ryanodine receptor could be instrumental in characterizing the skeletal muscle cells. Next, we have gap junction labeling, which is another method for characterizing our cardiac muscle cells. So, the gap junctions, we have discussed about the gap junctions in some of our previous lectures. So, these are basically connections within adjacent cardiac muscle cells, which are used for a quick propagation of electrical impulse through the cardiac muscle cells. So, antibodies directed against these gap junctions could help us in identifying the cardiac muscle cells in culture. Next, we have MHC identification or myosin heavy chain identification and calcium wave imaging, which are also some of the other techniques which are used to determine or ensure that we are growing the right kind of cell, or that our cell is mimicking the original in vivo condition in our in vitro state. So now we will move on to discussing the assignment questions. So question number 1 is which of these can be used to separate the population of two cell types from a mix? Immunopanning, fluorescence assisted cell sorting, both immunopanning and fluorescence assisted cell sorting, or none of the above. So you can write the answers in the chat box and we will discuss the answer. So all of you are saying that it should be option C. So this is one question that we had already discussed in the beginning of our slide. So immunopanning and fluorescence assisted cell sorting are two techniques that are used to separate the population of two cell types from a mix. Apart from that, we also have density gradient centrifugation, which is not included in our in the options here. So it should be option C, that is immunopanning and fluorescence assisted cell sorting. Moving on to question number 2. Which of these is a signature for all leukocyte groups? CD45 plus, CD45 minus, CD34 plus, or CD340 plus, or CD340 minus? So these are all examples of markers which are found on the surface of cell membrane, and they could be used to characterize our different cell type. So as we had already discussed that different cell types uh, express different kind of markers at different stages of their the developmental different developmental stages. So if we have a good knowledge of what kind of receptors are expressed on the surface of cell membrane at any particular time, we could use specific antibodies directed against them to separate them out from a mixture of cells. So which of these markers are expressed? Which of these markers are an example of a leukocyte marker? So all of you are saying that it should be CD45 plus. Let's see. So the right option is CD45 plus. So CD is basically an abbreviation for cluster of differentiation. CD molecules are cell surface markers which are useful for the identification and the characterization of leukocytes and the different subpopulation of leukocytes. And it is a transmembrane protein tyrosine phosphatase which is located on the surface of most hematopoietic cells. So as you can see in this figure given here, so this is here in this experiment we have used a certain antibody which is conjugated with a fluorescent which is conjugated with a fluorescent dye. So the here we have used the antibody directed against CD45. So we have used an anti-CD45 antibody that is conjugated with this dye, the APC CY7A. So, and now this we are measuring along the x-axis, and along the y-axis we are measuring the side scatter. So, as we had discussed before, that side scatter basically measures or gives us an implication about the granularity or the complexity of the cell. So, here we have obtained our result. We can see several cells here. So CD45 plus means that along the x-axis the cells which are CD45 plus, that is they bear the cells which have a receptor, a CD45 receptor on the surface, they will show up in this quadrant of the x-axis, that is more towards the positive side, this side of the x-axis, while along the y-axis we are measuring the side scatter, that is cells which are less granular or less complex will show up in this quadrant, the lower quadrant, whereas cells which are highly complex or granular will show up in the upper quadrant. So since we are selecting the leukocytes, we are trying to select the leukocytes from amongst this cell population, we will select the cells which are more situated more towards the this uh, right side of the x-axis. So we select the cells that we perform a gating. This phenomenon is known as gating. So gating is a method that we use in FACS. So this is done to tell our machine that we want to analyze this batch of cells further and stop analyzing the rest of the cells. So we will only take these cells which are situated towards the right side of our x-axis, that is which are CD45 plus, and we will take these cells to analyze further while we will stop analyzing the rest of the cells. Now again, similarly, from amongst these cells that we have selected here, we will take these cells and then we will again perform a second gating. So now we are selecting the cells for those we are selecting for those cells which express the CD3 marker on their surface. So again, similarly, we are measuring the CD3 marker on the surface, and we have taken the anti CD3 antibody, and then we have conjugated with some other kind of with some other fluorescent dye, and then again we are gating this, and then we are selecting for these cells which express the CD3, which bear the CD3 receptor on the surface. So similarly, next we are selecting for the cells which are CD4 positive and CD8 positive. So this way we can uh, select for the cells which are of interest to us with the help of FACS and with the help of gating. So question number 3 is a fill in the blank type question. So an blank is a protein molecule that recognizes a blank on the cell surface. Again, we have four options here: antigen, antibody, antibody, antigen, 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 or antibody, antibody. So you can write the answers in the chat box. So as we had seen in our previous question that antigens are expressed, antigens which are also referred to as markers are expressed on the surface of the cell membrane, and we use specific antibodies which are used to recognize and bind to these antigen and identify and sort them from the cell mix. So we will see what the answer is. So the answer is an antibody, that is option B, is a protein molecule that recognizes the antigen on the cell surface. So the function of the antibody involves specific binding to antigen and the activation of other components of the immune system to fight pathogen. So this is the typical structure of an antibody molecule. They are composed of two heavy chains, the longer chains, and two light chains. So these heavy and light chains are connected with the help of interchain and intrachain disulfide bonds. This region of the antibody molecule is associated with antigen binding. It is composed of both the heavy and the light chain, while this region, which is composed of the heavy chain only, is associated um, with mediating biological activity or effector activation. Next, uh, question number 4. This is again a fill in the blank type question. So in blank, there are two types of motor neurons. One are small in size called blank, and another large in size called blank. So you can write the answer again in the chat box and we will discuss. So in our previous lecture, we had discussed about the alpha and the gamma neurons. So we had discussed that the gamma neurons are used to innervate the interfusal fibers, and the alpha neurons are innervate the extrafusal fibers. Now you can write the answer and we will see what the correct answer is. So all of you are saying there should be option D. Let's see. So option D is the right answer. In ventral horn, there are two types of motor neurons. One are small in size, which are known as the gamma neuron, and other, which is larger in size, is called the alpha neuron. So this is the basic structure of a spinal cord. So in the adjacent figure, you can see the transverse section of the cord at three different levels: the cervical, the thoracic, and the lumbar sacral, which is not shown. So here in the transverse section of the spinal cord, you can see this their internal structure. This is somewhat the white matter and the gray matter somewhat arranged in this way. The gray matter is situated at the center, while the white matter surrounds it. So in transverse section, the gray matter is divided into the dorsal, lateral, and the ventral horns. So the neurons of the dorsal horns are associated with receiving sensory information that enters the spinal cord via the dorsal root to the spinal nerve, while the ventral horns contain the cell bodies of motor neurons which send their axons via the ventral root of the spinal nerve to terminate on striated muscle. So the dorsal horns are associated with receiving information, while the ventral horns are associated with sending information to the striated muscle. While the lateral horns are present primarily in the thoracic region, and they contain the preganglionic visceral motor neurons that project to the sympathetic ganglia. The white matter of the spinal cord is subdivided into dorsal, lateral, and ventral columns, each of which contains axon tracts related to specific function. The dorsal columns carry ascending sensory information from somatic mechanoreceptors. The lateral columns include axons that travel from the cerebral cortex to contact the spinal motor neurons. The ventral columns carry both ascending information about pain and temperature and descending motor information. Some of the general rules of spinal cord organization are that the neurons and axons that process and relay sensory information are found dorsally. The preganglionic visceral motor neurons are found in an intermediate region, while the somatic motor neurons and axons are found in the ventral portion of the cord. Next, we have question number 5. In rat spinal cord at E14, only motor neurons express a particular marker. This information can be used to isolate the cells from mixed culture with other cells of the spinal cord, test the electrical activity of these cells, digest these cells, or none of the above. So we have seen that markers are used to identify cells at a particular stage. We could use specific antibodies directed against them to identify cells bearing a particular kind of marker. So you can write the answer and we will see what the correct answer is. So all of you are saying there should be option A. So let's see. So yes, the right option is A, that it is used to isolate these cells from mixed culture with other cells of the spinal cord. Next, moving on to question number 6. Trypsin is used to do what? By digesting the extracellular matrix, as it is a, again we have a blank. So there are 4 options here. lift cultured cells off a surface condensation enzyme, lift culture cells of a surface, proteolytic enzyme, attach cultured cells of a surface, condensation enzyme, or attach cultured cells of a surface, proteolytic enzyme. So some of our early sessions we had discussed about the basics of cell culture, and there we had discussed about the enzyme trypsin, which is used to degrade the cell-cell interaction, cell matrix interaction, and obtain a single cell suspension of cells. Or so we use trypsin to detach the cells from growing on the surface of our culture plate and then we um, obtain a single cell suspension of our cells, which are then used to say if we are performing a passaging of the cell, we use these trypsin to detach the cells off the surface of a growing on the surface of a plate, and we use the enzyme trypsin to detach the adherent cells which are growing on the surface, and then we bring them into a single cell suspension, and then we passage them. So all of you are saying that it should be option B. Let's see. So the option is B. The right answer is lift cultured cells off a surface, and it is a proteolytic enzyme. So it is used to break the different protein-protein interaction, the different protein molecules it is used to break the protein-protein interaction between different cells of the cell and the matrix. Next, moving on to question number 7. Uh, which of the following is a supplement used for serum-free fetal hippocampal neuron culture media? B27, G5, collagen, or fibroin? So we discussed about this in some 2 to 3 classes ago. So which of the following is a supplement that is used for serum-free fetal hippocampal neuron culture media? So a couple of you are saying there should be option A. Let's see. Yes. So option A, that is B27, is the right answer. B27 is a neuronal cell culture supplement. It is an optimized serum-free supplement used to support the low or high density growth and short or long-term viability of embryonic, postnatal, and adult hippocampal and other central nervous system neurons. B27 is also used as a supplement for the culture of our mammosphere cells. Next, question number 8. This is also somewhat related to question number 7. Which of these is not a component of B27 supplement? Vitamin A, biotin, cutase, or sodium selenite? So you can write the answer and we will see what the correct answer is. So a couple of you are saying should be option C. So let's see. So yes, option C, that is acutase, is the right answer. So these are the components which are generally present in B27. We have biotin, we have vitamin A, and we have sodium selenite. Apart from this, we also have these other vitamins and proteins and components which are present. And depending upon the manufacturer that is uh, producing the supplement, these components can be varied to contain or uh, not contain some or few of these components. So acutase is the right answer. Now moving on to question number 9. So Glutamax is what and has a blank than glutamine. So we have 4 options here: L-alanine, L-glutamine, lower stability, L-alanine, L-glutamine, higher stability, L-glycine, L-glutamine, lower stability, L-glycine, L-glutamine, higher stability. So you can write the answer. So we have discussed a similar question in some of our early sessions. You can write the answer and we'll discuss. So you're saying there should be option B. Let's see. So option B is the right answer. Glutamax is L-alanine L-glutamine and has higher stability than glutamine. So although L-glutamine is an essential nutrient in cell culture, it spontaneously degrades in cell culture media, generating ammonia and pyrrolidine carboxylic acid as byproduct. The glutamax supplement is an improved cell culture supplement that can be used as a direct substitute for L-glutamine in your cell culture media. Glutamax supplement is a dipeptide L-alanine L-glutamine, which is more stable in aqueous solution and does not spontaneously degrade. Next, the last question, question number 10. So, read the following statements carefully. Animal origin free medium, we have four statements here, is not manufactured using any raw material ingredients derived from an animal, including human, tissue, cell, or body fluid, that is at the primary level of manufacturing. No animal-derived materials are used as a starting material or in the primary level manufacturing process. Statement B, the components used to manufacture the raw material ingredients are not derived from an animal, tissue, cell, or body fluid, that is at the secondary level of manufacturing. Statement C, ingredients and components may be produced by or extracted from plants, bacteria, or yeast or cell-free systems derived from them, or may be produced by organic synthesis. And statement D may contain recombinant animal or non-animal proteins provided they are expressed in plants or microorganisms. So again, we have four options here. Only statement A is true, only statement B is true, only C and D are true, or A, B, C, and D are all true. You can write the answers. You're saying there should be option D. It is all of the above statements are true. So let's see. So yes, option D is the right answer. Uh, so that is A, B, C, D, C and D, all of the statements are true. So depending upon how each supplier define their culture media, the cell culture media could be either of different kinds. They could be chemically defined, or it could be protein free, animal component free, xeno free, or serum free. So animal origin free means that the finished product does not contain or use in the manufacturing processes any primary and secondary raw materials which are derived directly from animal, including human, tissue, or body fluid. The tertiary raw materials may be derived from the animal, including human, tissue, body fluid, or cell line. That was the last question. Now we have a few additional questions here that we have added. So the first question is in flow cytometry, what is the side scatter a measure of? Granularity, density, size, or fluorescence intensity? So you can write the answers in the chat box and we will discuss. So all of you are saying there should be option A, that is granularity. Let's see. So yes, option A is the right answer. The side scatter is a measure of the granularity, whereas the forward scatter is used to measure the size of the cells. Next, we have the next additional question. In a flow cytometer, the particles are analyzed all at a time. So is this statement true or false? You can write the answer in the chat box. So all of you are saying that it should be option B, that is false. So let's see. So yes, this statement is false because in flow cytometer, the hydrodynamic focusing of cells ensure that they are made to pass in a single file, that is one by one, through the nozzle, so that they are interrogated one at a time, and the results are interpreted one at a time. So the particles are not analyzed all at the same time, and therefore the statement is false. Next question. So this is a bit of a tricky question. So let's see if we can answer this. So in human, normal B and T cells develop the CDC4 plus and CDC8 plus first, and then they differentiate to CDC4 plus, CDC minus, and CDC4 minus, CDC plus. So if there is a defect in the development, which quadrant in the flow cytometer graph will show more density? So we have four quadrants here: 1, 2, 3, 4. So suppose along the x-axis we are measuring the see we are measuring the CDC4 plus marker, and along the y-axis we are measuring the CDC8 plus. And so the question tells us that we have we in the normal human B and T cells, they initially during the development process they express both of these markers, that is they express both CDC4 plus and CDC8 plus. However, as they develop further, they differentiate, that is initially both B and T cells will look the same. Both of them bear the CDC4 plus and the CDC8 plus marker. However, uh, after uh, after a while they differentiate to become CDC4 plus, CDC8 minus, and CDC4 minus, CDC plus 8 plus. So if there is a defect in development, which quadrant in the flow cytometer graph will show more density? So let's see if you can answer this question. So you have four options: 1, 2, 3, and 4. So you can write the answers in the chat box, and then we will discuss what should be the right answer. This is a bit application based question based on the facts technique. Let's see if we can answer this question. So one of you is saying there should be option C. So let's see the right answer. So the right answer is option 2. So what we are trying to understand here is that initially, the question says that initially when the B and T cells develop, they express both these markers on their surfaces. That is, they express both the CDC4 plus as well as the CDC8 plus markers on their surface. So, so if we are measuring the CDC4 plus marker along the x-axis, as we had seen in one of the questions related to the CD45 plus marker. So if we are measuring the CDC4 plus marker along the x-axis, and we are measuring the CDC8 plus marker on the y-axis, so CDC4 plus cells which bear the CDC4 marker should appear towards the right hand side of the x-axis, while cells which are right towards the sides of the cell of the x-axis, and cells which are CDC8 plus, that is they bear the CDC8 plus mark on the surface, they should be expressed towards the upper portion, that is towards the upper portion of the y-axis. So during the initial developmental condition, since both these markers are being expressed, so we can expect that they will be more expressed in this region, that is the quadrant 2, because since they are both these markers are being expressed. So now they are saying that if there is a defect in development, which quadrant in the flow cytometer graph will show more density? So so as they begin to develop further, one of the B cells, or say the T cells, they develop these characteristic phenotype, that is they lose one of their marker. So either they are CDC4 plus and CDC 8 minus, or they are CDC4 minus and CDC8 plus. So if they're CDC4 plus and CDC8 minus, we can expect them in if they're CDC4 plus, that is if we can expect this that they will be in this region. And if they're CDC8 plus and CDC4 minus, we can expect them in this region. So if there is a developmental problem happening which prevent them from differentiating, we can expect them to retain their original or their initial or their phenotype. So they will be in this region, that is they are expressing both the markers, and we can expect them to be in the quadrant 2. So this was an application based question of facts. Next, moving on, uh, which of these statements is not true regarding skeletal muscles? They are also called voluntary muscles. They help in the movement of food through the esophagus. They have striations, and they are involved in changing of body posture. So you can write the answer in the chat box and we will see. So you are saying there should be option B. That is, they help in the movement of food through the esophagus is the wrong statement. So let's see. So yes, that is the wrong statement. So skeletal muscles are also known as voluntary muscles. They are striated and they are involved in changing of body posture. However, they do not help in the movement of food through the esophagus. They are this is a function of the involuntary muscles, which are not under our self control. So this is not a function of the skeletal muscles or the voluntary muscle. Next, which of these is not a characteristic of cardiac muscles? That is, they work continuously, they are branched, they are not striated, or they are involuntary. You can write the answer. It's also probably a last question. So the cardiac muscle cells are bare striations, they work continuously, and they are not are under our voluntary control. Let's see what the right answer should be. So option B is the wrong statement, that is, they are branched, because cardiac muscle cells are not branched. Rest of the options are correct. That is, they work continuously, they are not striated, and they are involuntary. So that's all for today. I would like to thank NPTEL and IIT Madras for giving me this PA-ship opportunity. I would like to thank Professor Moinav Das, the course instructor, for this course for selecting me for this role. I'd like to express my gratitude to my supervisor, Professor Mahitosh Mandal, sir. I would like to thank MHRD Government of India for the Prime Minister's Research Fellowship. I thank IIT Kharagpur for allowing me to pursue PhD at the institution. And lastly, I thank all the participants for joining the session. Thank you.